The nephron is the functional filtration unit of your kidneys. Now, I mentioned it in previous videos, but we really need to come back and talk about what it is, the parts, the pieces of it, and how it works. So we'll start with going through some of the anatomy of it at this point. Now, what's interesting though is you have about, give or take, 1 million of these nephrons per kidney. That just gives you an idea of how many of these things are packed into your kidneys. Now, there's two main components. You have the glomeralis. The glomeralis itself is capillaries. This is how the blood gives the filtrate, or the components that need to be filtered to the kidney. Then the rest of it is the renal tubule. Now, this renal tubule is going to have different curves, different parts, different functions. But we're looking at the start. The glomeralis is the capillaries. And the glomerular capsule is what's around it. So you're going to have the glomeralis, which is blood vessels, and then the tubule around it, the capsule. So that's the basis of how this is starting off. Now we need to look at the actual structure of a nephron. Now you have one nephron here, and it's shown just coming out of that little tiny box over there. So really you have these all scattered throughout the entire cortex. So the first thing to look at is where the cardiovascular system, the blood, interfaces with the nephron. And that's at this thing called the renal corpuscle. Now the renal corpuscle has two main components. You have the glomeralis, which is the capillaries inside, and then you have the glomerular capsule, which is what is the outside. Together, the capillaries inside and the capsule outside make the renal corpuscle. So glomeralis, capillaries, glomerular capsule outside, together is the renal corpuscle. Coming off of that, you have this proximal convoluted tubule. Proximal just means closer to. Convoluted because it's all curving and winding around. Now in this image, they show it being curved, but they show it in a very nice, neat, compact aspect. In reality, that's curving around every other tube. They're all mixed together. They're trying to oversimplify this so it's easier to see the parts. But just keep in mind, there are all these different nephrons, all side by side, all interweaving together. So the proximal convoluted tubule is going to take the filtrate that was collected in the renal corpuscle. It'll then take that filtrate, go through the proximal convoluted tubule, heading down into the loop of Henle, or the renal loop. Now notice the terms like thick and thin segment. You can see how this one here is thicker and this area is thinner. It's physically what you're looking at. Then you also have the descending and ascending limbs. Well, as you go from the proximal convoluted tubule, you go down the descending limb, you come back up the ascending limb. So two different parts that go into this loop of Henle. From there, we continue on to the distal convoluted tubule. Distal meaning further away from. It's further away from the renal corpuscle. Now, each one of these areas, distal convoluted tubule, loop of Henle, proximal convoluted tubule, the renal corpuscle, they each have their own purposes and functions. So keep that in mind. Eventually, we'll leave the nephron and could go into the collecting duct. But let's start with that renal corpuscle first and kind of work our way back through it. So again, the renal corpuscle, we already mentioned, glomeralis and glomerular capsule. You're going to have this fenestrated glomerular endothelium. Remember, fenestrated means it has more openings. That's allowing for the filtrate to pass from the plasma of the blood into the renal corpuscle. So into that capsule there then. Because you have to get things out of the blood so they go into the renal tubes. So that's what we're looking at for this renal corpuscle, the starting point, how it leaves the blood, goes into the nephron. Now the glomerular capsule itself has a parietal layer that is simple squamous epithelium. Makes sense, nice and thin, easily permeable, allowing things to pass through. But we also have the visceral layer. Now this visceral layer is going to have branching what are called podocytes. Podocytes are specialized cells that help give structure to the membrane. They're around the membrane. So it's helping to give a little more stability to this. But we have these filtration slits in between the podocytes. So the podocytes kind of form these interlocking aspects. You have these slits in between that aid and help with the filtration from the plasma of the blood into the filtrate inside the nephron. So what we're looking at here, again, same picture of the actual nephron, but now you'll notice the glomerular capsule, parietal layer, simple squamous, very thin. You then go down to the glomerular capsule, visceral layer. You can see this is the podocyte that is, there's almost membrane on top of it. It's helping to kind of hold and support, creating these filtration slits. So some unique aspects there, especially the podocytes, they're wrapping around and the parietal and the visceral layers here. So parietal layer, simple squamous, visceral layer is going to be the podocytes. Moving on to the proximal convoluted tubule, PCT, proximal convoluted tubule. You'll notice you're going to have cuboidal cells. They're going to have microvilli helping to kind of move things along here. One of the main functions here is reabsorption, but also secretion as well. Because all this filtrate going into the nephron, you're not going to have all this being excreted out of your body. You need to reclaim a lot of that back into your blood. So there's a lot of reabsorption going back. So things that were filtered out are being put back into the blood. Because you only want to keep things in the kidneys that you want to get rid of. You're not getting rid of this much filtrate. Now these proximal convoluted tubules are always found in the cortex. You're not going to find these in the medulla. But the loop of Henle, that can be found in the cortex and down in the medulla as well. The thin segment, a lot of times, simple squamous epithelium, it's usually part of the descending limb. So as you're going down the loop of Henle, that's where you're freely permeable to water. So water can be easily exchanged and be pulled back into the system. Because remember, you're trying to conserve what you have. You're not trying to have yourself drink thousands of milliliters of water in a day. Because you simply can't drink that much. So you've got to conserve your water. Here, the system's pulling it back out so it's not being excreted. The thick segment usually is in the ascending limb. Now this one, cuboidal, columnar cells, a little bit different structural setup here. We can still have some water, but it's not as freely permeable. Really, that descending limb really is very permeable. Then we have the distal convoluted tubule. In the case of the distal convoluted tubule, again, we're confined to the cortex, so we're only up on the cortex. Now, there's not many microvilli here, so it's not having the same functionality. Now, this one is more secretion-based than reabsorption. So a little different thing we'll talk about more as we get later, but it's more secretions instead of reabsorbing. And then those collecting ducts I briefly mentioned as we left the last picture of the actual nephron. Collecting ducts are what each nephron goes into. The collecting duct is going to basically have many nephrons all joining, and all that filtrate is heading down. It's going right into the papillae of the pyramid. So remember when you have the pyramids, the very tip, the end was the papillae. That goes in the minor calyx. Minor calyx goes in the major calyx. That is the urine at that point. So we're looking at all these nephrons joining together to make these collecting ducts. Now the collecting duct has two main cells to look at. We have these intercalated cells. The intercalated cells have microvilli. They're going to be dealing with the acid-base balance of the body. So helping to further that homeostatic balance of acid-base. Try to make sure you keep the blood at that 7.35 to 7.45 range. Making sure it's right where it needs to be. The other cells are principal cells. Now these principal cells deal with the body's water and salt balance. Now you say, well, water and salt, those are two different things. They go hand in hand. Because if you retain salt, sodium chloride, water wants to follow because you want to dilute the salt. So both salt and water balance makes sense to go together. And that's now these principal cells. So again, looking at the different tissue types, you'll notice that the proximal convoluted tubules that are located up in the cortex, they have plenty of microvilli on their surface. But then you get to the loop of Henle. Loop of Henle, thin segment there, a little bit flatter cells. But then the distal convoluted tubule, notice there's not as many microvilli. We've decreased the numbers. And then the collecting ducts, you have two different types of cells here. You have the principal and the intercalator member. So looking at some of the histology of the nephron as well as the nephron structure itself. So the nephron itself, structural components, and a little bit about what they're doing. We definitely need to talk more about the physiology of how this all works though.